Welcome back to the deep dive. This is where we take essential texts and really break them down for you, giving you that critical knowledge fast.
That's right.
Today, we're tackling something absolutely foundational for any surgical career. Not necessarily the knife skills, but the intellectual rigor behind it all. We're talking research, audit, quality assurance.
Yeah, absolutely. Our goal today is to give you a really solid, deep understanding of that whole landscape, how surgical inquiry works, from question to, well, maybe publication. And we're sticking just to the core principles you need.
Because it really is a cornerstone, isn't it? You need this stuff for generating evidence, obviously, but also just for meeting professional standards like revalidation.
It's completely fundamental. And for you, the surgical learner, maybe revising for exams or just starting out, the whys is huge.
Totally.
This isn't just about ticking boxes. It's about developing those critical appraisal skills.
Yeah.
You're going to be hit with a flood of new papers, new studies every single day.
Right.
If you don't understand how a good project is built, how research is actually done properly, you can't really judge the findings effectively, can you?
You're just sort of taking it at face value.
Exactly. And we often find, the hardest bit isn't always the idea or even collecting the data. It's writing it up or even just figuring out where to start.
Yeah, the blank page fear.
Precisely. So today we want to give you that structure, the framework you need to look at any paper or start your own project and feel confident about it.
Okay, sounds good. Let's dive in. All right, first things first. Before you even think about patients or data collection, you have to classify what you're doing. Is it audit or is it research?
And this isn't just semantics. This decision dictates everything that follows. The rules, the approvals, the funding pathways. It all hinges on this first step.
So what's the core difference then?
It really boils down to quality assurance versus generating new knowledge.
Right.
Clinical audit and also service evaluation, they're essentially QA activities. Quality assurance. Things that, health professionals are actually expected to do as part of ensuring good care. It's about checking how you're doing things now.
Okay.
So usually there's minimal extra risk or burden involved because you're mostly just looking at existing practice against a known standard.
Let's nail down that definition of audit then. You said known standard.
Yes, clinical audit is a systematic process. It's about quality assurance. You compare aspects of care, could be the structure, the process, the outcome, against explicit criteria and defined standards.
And those standards, they already exist. You're not inventing them.
Correct. They must already exist and be agreed upon. Maybe from national guidelines or local protocols. You're measuring yourself against the benchmark.
Which brings us to personal performance, right? The text emphasizes this.
Hugely important. Every surgeon needs to be constantly monitoring their own outcomes. Things like mortality, complications, how long patients stay in hospital. And contributing that data, usually anonymized, to local or national databases. Comparing your results against national benchmarks is absolutely vital. It's a mandatory part of revalidation for surgeons in the UK, for instance. It shows you're maintaining standards.
Okay, so audit is measuring against an existing standard. How do we draw the line? When does it tip over into being actual research?
Well, the UK's Health Research Authority, the MHRA, they developed a really useful decision tool. It basically comes down to 3 big questions. Anyone involved in this needs to know these.
What are they?
Okay, the first 2 are pretty direct. They're about what you're doing to the patients. Question 1. Are participants randomized into different groups, like in a drug trial?
Right.
Question 2. Does your study plan require changing the treatment, the care, or the service away from what's considered the accepted standard for any of the patients?
Okay, so if you answer yes to either of those...
You're immediately in research territory, no question. You'll need formal research, ethics approval, governance sign-offs, the whole package.
But there's a third question, and you said this one can be tricky.
Yeah, this is often the one that causes confusion. The question is your study designed to produce generalizable or transferable findings?
Generalizable. Okay, what does that actually mean in this context? It sounds broad.
It is. And the HRA definition is key here. For findings to be generalizable, it means you should be able to reliably extrapolate them from the people in your study to a wider population of patients.
Wider population.
Or apply those findings reliably in different places, different settings, beyond just where you did the study.
Okay, let me test that. Say I'm a junior doctor and I'm just looking at how quickly emergency surgeries happen in my hospital. Are we hitting the 2-hour target, for example? That doesn't sound generalizable, is it?
Probably not, and that's the crucial distinction. Your local audit might find, say, a bottleneck in getting patients to theater between 2 and 4 a.m. That finding is likely specific to your hospital's staffing rotas, maybe your portering system, your specific theater access protocols. You can't just assume the hospital down the road has the same problem, or that your solution, maybe changing the on-call rota, would work there.
So it's local information, not universal truth.
Exactly. So most local audits like that are considered hypothesis generating. They might suggest an idea, maybe staffing is the issue, but they don't provide new established knowledge that's proven to apply elsewhere.
Got it. So they don't meet that strict test of creating knowledge that changes practice everywhere?
Precisely. Now, sometimes you hear the term transferable findings, often used more in qualitative research like patient interviews.
Okay.
That means the insights might be considered relevant or applicable in a similar context or setting, even if not strictly generalizable everywhere. But the moment your primary aim is to create new knowledge that you intend to be widely applicable, you've crossed into research.
Okay, let's stick with clinical audit for a bit. You said it's a formal process, not just ticking boxes.
Definitely not. It's a continuous cycle, really. And it needs structure. The text lays out a classic 7-step audit cycle. It's essential.
Walk us through it. Step 1.
Step 1. Define the audit question. What exactly are you measuring? And importantly, do this with a multidisciplinary team if possible. Surgeons, nurses, maybe physios, anesthetists. Get everyone involved who touches that pathway.
Makes sense. Step 2.
Identify the evidence and the standards. What's the benchmark you're measuring against? This has to be an agreed, established standard, like we said, maybe a nice guideline or a Royal College recommendation.
Okay. Step 3.
Design the audit itself. How are you going to measure performance against that standard? What data do you need? Who will collect it? How? You need a clear methodology. And crucially, get agreement from your hospital's audit department before you start collecting data.
Right. Governance first. Then steps 4 and 5 seem straightforward.
Yes, step 4 is just doing it, measure performance over an agreed time period. Step 5 is crunching the numbers, analyze the results, and compare your performance to that standard you set back in step 2.
Okay, now step 6 sounds like the crucial point, the gap analysis.
It really is. This is where you make a decision based on the data. If you're meeting the standard, fantastic. Schedule a re-audit for maybe a year's time just to check things are still on track.
But if you're not meeting the standard, if there's a gap,
then you need to figure out why and what you're going to do about it. You need to identify specific interventions to improve performance.
what kind of interventions are we talking about? It can't just be try harder, right?
Absolutely not. It has to be concrete. It could be relatively simple, maybe some specific training for staff who aren't following a protocol. Or maybe it's about resources. Perhaps you need different equipment like upgrading laparoscopic sacks.
Okay.
Or it could be more complex, requiring bigger changes. Maybe implementing a whole new preoperative checklist, changing theater scheduling patterns, or even needing to make a case for more regional funding for a specific service.
An agreement is key.
Essential. Whatever the intervention, the people who actually have to do it need to agree it's feasible and necessary. Otherwise, it just won't happen.
And the final step, step 7.
Re-audit. You have to close the loop. After you've implemented the changes from step 6, you wait a suitable period, and then you run the audit again, using the same methodology.
To see if the changes actually worked.
Exactly. Did performance improve? Did you meet the standard this time? If yes, great. Cycle complete for now. If not, you might need to rethink your interventions and go around again. It's a continuous improvement process.
Okay. That cycle makes sense for, say, a single hospital department. But you mentioned earlier that single-site stuff can lack power.
Yeah, especially in surgery. If you want to look at variations across the board or generate really robust hypotheses, a single hospital audit might not cut it. That's where these multi-center snapshot audits have become really popular.
Right. You mentioned these gaining traction in the UK and Australia. What's the setup?
Instead of one hospital collecting data slowly over months, you get lots of collaborators, often trainees, across many different hospitals.
Okay.
They all agree to collect specific anonymized patient-level data on a particular condition or procedure, but just for a short, defined window of time, often just 6 or 8 weeks, like a snapshot in time across the whole system.
What are the advantages of doing it that way?
Oh, they're huge. Firstly, they're really accessible. Junior doctors and trainees can easily get involved. The cost is minimal because you're usually using existing hospital systems and staff time. They're a fantastic way to build collaborations between hospitals. And critically, you get a large volume of real-world data very quickly, showing what's actually happening now
Yeah.
across many different places.
So they're not really trying to change the standard immediately, more just map the landscape.
That's a perfect way to put it. The main goal is usually to explore practice variability. How are 50 different hospitals managing acute appendicitis, for example? Are there big differences in antibiotic use or imaging strategies or operative approach?
And seeing that variation.
And correlating it with outcomes, even basic ones like length of stay or readmission rates. That can generate really powerful hypotheses. It tells you where the inconsistencies are and which variations might be worth investigating properly in a future, more formal prospective research study, maybe even an RCT. It's like doing the groundwork.
Oh, okay, so now let's shift gears into actual research. Generating genuinely new knowledge. This demands, well, a whole different level of rigor right from the very beginning, starting with the idea itself.
Right. You've got maybe a clinical puzzle, something you've noticed isn't quite right or could be better, but that needs refining, doesn't it, into a proper, testable hypothesis.
Absolutely, and the text is really strong on this. Spending enough time sharpening that clinical question, making it precise, is arguably the single most important part of the whole research journey.
More important than the fancy stats later on.
I'd argue yes, in many ways. Because if you start with a flawed or vague question, everything that follows is built on shaky ground.
There's a warning here, too, isn't there, about rushing in? A big warning. Don't just jump into designing the study. The worst possible outcomes, and they happen, are spending potentially thousands of hours and maybe significant money only to find out, oh, someone else already definitively answered this question 5 years ago.
Ouch.
Or, arguably worse, you get to the end, you've collected all your data, and you realize your methodology was fundamentally flawed. It just couldn't answer the question you set out to ask. That's incredibly demoralizing.
So how do you avoid that? How do you find that sweet spot? A question that's important, unanswered, and actually feasible to investigate.
Well, the first stop is usually the literature. The internet is your friend here, obviously, but medical librarians are absolute gold. You need to find the most current, systematic reviews and meta-analyses in that specific area.
Because they summarize everything.
Exactly. They explicitly state what we know and often what we don't know, where the evidence runs out. But, and this is a common pitfall, it is not enough just to read the abstract.
Guilty as charged sometimes.
We all are. But you have to read the full papers, especially the discussion and limitations sections. That's where the authors often highlight the remaining uncertainties.
Okay, beyond reviews, where else can we look for good research questions?
The text points to a really high-yield source. High-quality national or international clinical guidelines. Think UK NICE guidelines, for example.
How do they help?
Most good guidelines have a specific section, often right at the end, labeled something like Recommendations for Research.
Okay.
That section exists because the expert panel who wrote the guideline reviewed all the evidence and identified specific gaps questions they couldn't answer definitively. If you design a project that directly addresses one of those questions, well, it's very easy to justify its importance to ethics committees, funders, and your colleagues.
That's a really practical tip. Any other structured ways?
Yeah, there are things called priority-setting partnerships. Groups like the James Lind Alliance often facilitate these.
What do they do?
They bring together patients, their carers, and healthcare professionals. And collaboratively, they work out what everyone thinks are the most important unanswered questions in a particular field, say managing arthritis or recovery after stroke. They often publish a top 10 list of research priorities.
So research aligned with those is likely to be seen as really relevant.
Extremely relevant, yes. And then there are support networks. Don't feel you have to figure out the perfect design all by yourself.
Like who?
Well, in the UK, for example, the National Institute for Health Research, the NIHR, runs the Research Design Service, or RDS. They offer free expert advice on study design, statistics, funding applications, even before you submit anything. Definitely worth using if it's available to you.
Okay, so you found your gap, refined your question. Now the team, you said earlier, this is where things can really fall apart.
It's often the make or break factor, honestly. Meaningful research is very rarely a solo effort. Almost any project worth its salt needs a team.
Why? Just for workload?
Workload is part of it, sure. But it's
Yeah.
more than that. You need diverse skills. Maybe someone's great at stats, someone else at recruitment, someone else has deep clinical expertise. You need different perspectives,
Yeah.
experience.
Okay.
But maybe the most critical thing a team provides, momentum. Research always hits roadblocks. Clinical pressures mount, exams come up, people move jobs. A good team keeps the project moving forward through those inevitable hurdles. One person alone can easily stall.
And surgery has a particular model for this, doesn't it? The trainee collaborative.
Yes. Surgery really pioneered this. The prototype was the West Midlands Research Collaborative, the WMRC, started back in 2008.
And the core idea was?
It was quite brilliant, really. Surgical training involves rotating between different hospitals, right? Which normally makes long-term research tricky for trainees.
Yeah, you move just as you get started.
Exactly. The WMRC flipped that. They used the rotation system as a strength. By having a network of trainees across lots of hospitals in the region that could ensure project continuity even as individuals moved, it created a large, stable pool of collaborators.
Clever. Did it work?
Amazingly well. The proof of concept was the ROSSINI trial. This was the first big randomized controlled trial run entirely by the WMRC.
What did it look at?
Something quite simple and practical. A wound edge protection device to reduce infection after surgery. But the key was the execution. The trainee network mobilized 21 different hospitals. They randomized 760 patients in just 23 months. Finished ahead of schedule.
Wow. That really showed the power of the model.
It absolutely did. It proved that trainees working collaboratively could deliver high-quality, large-scale research. It wasn't just about numbers. It was about using that rotational structure strategically.
That's a fundamentally different way of thinking compared to the old model of maybe one consultant doing a study in their own hospital.
Totally different. And that success just sparked an explosion. Now you have similar trainee-led collaboratives all over the world. StarSurg is another big one in the UK. There are groups in Canada, Australia.
And not just surgery.
No, it's spread. Anesthetics, elderly care, other specialties are adopting the model too. And they're typically very inclusive. Medical students right through to consultants can get involved. They are genuinely changing how surgical evidence is generated using collective effort.
Okay, team assembled, killer question identified. Now we get down to the nuts and bolts. Designing the actual project, the blueprint.
Exactly. This is where you map out precisely how you're going to answer that question.
Yeah.
And there's a sort of pre-flight checklist you absolutely have to go through. Brutal honesty required.
What's on the checklist?
Things like, why are we really doing this? Is the answer actually going to be useful to anyone? Is it practical? Do we genuinely have the resources, the time, the money, the staff, the patients? What are the ethical hurdles likely to be? What regulatory hoops do we need to jump through? And ultimately, what impact could this study realistically have, even if it works perfectly? You need solid answers to all of those before you commit.
Okay, assuming it passes the checklist, selecting the study type. We hear a lot about randomized controlled trials, RCTs being the gold standard.
They're often considered the highest level of evidence for interventions, yes, because randomization minimizes bias. But surgical research is complex. We rely on a whole range of study designs.
Such as?
Well, there are observational studies. These can be retrospective, looking back at data that already exists.
Like patient records.
Exactly. Or they could be prospective, identifying a group of patients now and following them forward in time, collecting data as you go. There are also case control studies, comparing people with a condition to those without. Cross-sectional studies, looking at things at a single point in time. And longitudinal studies, which follow the same group over many, many years. Each has its strengths and weaknesses.
What about totally new surgical techniques? You can't always do a simple RCT straight away, can you? Like, you can't easily blind someone to whether they had keyhole or open surgery.
That's a really important point. Surgical innovation is tricky. The text mentions the ideal framework recommendations specifically for evaluating new surgical procedures or devices.
Ideal.
Yeah, it stands for Idea, Development, Exploration, Assessment, Long-Term Study. It proposes a staged approach. You start small. Phase 1 is just about safety the first few times it's done. Then you gradually expand, maybe comparing it to historical data, before potentially moving to a full RCT, comparing it against the current standard care, which would be phase 3.
So it acknowledges the learning curve and the unique risks.
Precisely. You have to evaluate innovation methodically and ethically. You can't just jump straight to a massive trial.
Let's talk data types. The text mentions balancing quantitative and qualitative data.
Yes, this is increasingly recognized as best practice. Quantitative is about the numbers, the hard, objective endpoints, and things you can measure precisely. Mortality rates, length of hospital stay, complication rates, and often we use standardized systems here, like the Calavian-Dindo classification, to grade surgical complications consistently. Blood loss measurements, things like that.
Okay. And qualitative.
Qualitative research is about understanding the experience, the narrative. It gathers data from patient interviews, focus groups, looking at the psychosocial impact of a condition or treatment, pain levels, depression scores, functional recovery from the patient's perspective.
And this is where things like PROMs come in, patient-reported outcome measures.
Exactly. PROMs are questionnaires that capture the patient's own assessment of their health, function, and quality of life. They're becoming incredibly important.
So the best research uses both?
Often, yes. A mixed methods approach, combining the objective numbers with the subjective patient experience, gives you a much richer, more complete picture of whether an intervention is truly beneficial.
Okay, let's tackle the big one, sample size. The text is almost alarming about this. It says an incorrect sample size is possibly the most frequent reason research is invalid.
It's certainly one of the most common and most critical flaws. If your study is too small, you simply don't have the statistical power to reliably detect a difference, even if one truly exists.
Which leads to errors.
Two main types. A type I error is a false positive. Your stats say there's a difference, a benefit, but in reality there isn't one. It happened by chance.
Okay.
A type 2 error is a false negative. There is a real difference or benefit, but your study was too small to pick it up reliably. You miss a potentially important finding. Getting the sample size right is about minimizing the risk of both these errors.
So how do you calculate it? What goes into the calculation?
You need to work with a statistician, ideally.
Yeah.
But the key inputs are usually the desired level of statistical significance, often set at 5% or p.05, and the desired power, usually set at 80% or increasingly 90%.
Kind of power. That's the chance of finding a difference if it's real.
Exactly. 90% power means if a true difference exists, you have a 90% chance of your study detecting it as statistically significant.
Okay, significance and power.
Yeah.
What else?
The crucial ingredient is the expected effect size. How big a difference do you realistically expect to see between your groups, based on previous research or pilot data, and what size of difference would actually be clinically meaningful?
So you need to estimate the benefit you're looking for.
Yes. For example, the text gives an illustration. If standard care has a 30% complication rate, and you hope your new intervention reduces it to 20%, that's a 10% absolute reduction.
Right.
To reliably detect that specific difference, 10%, with 90% power and 5% significance, the calculation shows you'd need about 333 patients in each group. So 666 patients total.
Wow, that's quite a lot. What if you were only expecting a smaller difference, say reducing complications from 30% to 25%, just a 5% drop?
You'd need a much larger sample size, maybe thousands. That's why you have to be realistic about the effect size. Trying to detect tiny differences can make trials impractically large and expensive. You need to focus on differences that would genuinely change practice.
And you also have to account for dropouts,
Right?
Attrition.
Always. People move away, they withdraw consent, they might unfortunately pass away from other causes during a long study. You must inflate your recruitment target to account for this expected loss to follow up. If you calculate you need 666 analyzable patients at the end, you might need to recruit, say, 750 or 800 to allow for 10–15% attrition. Otherwise, you end up underpowered.
Final point on design, bias. How do we minimize that? Blinding comes in here.
Yes. Bias is any systematic error that makes the results deviate from the truth. Blinding or masking is a key strategy. Single blind means the patient doesn't know which treatment they got. Double blind, the gold standard, means neither the patient nor the researcher assessing the outcome knows the treatment allocation.
But you said earlier double blinding is often hard in surgery.
It often is. You can't blind a surgeon to whether they're doing a laparoscopy or an open operation. It might be unethical or just physically impossible.
So what do you do then?
In those situations, you focus on blinding the person who assesses the outcome. For example, the radiologist reading the post-op scans, or the physiotherapist measuring functional recovery, or a research nurse assessing wound healing. They should ideally be unaware of which surgical group the patient was in. Using these blinded observers helps reduce assessment bias, even if the patient and surgeon couldn't be blinded.
Okay, we've designed the study. Now we need the instruction manual, the protocol. You called it the Bible of the trial earlier.
That's pretty much what it is. It's the single, detailed document that lays out exactly how the study will be run. It has to be incredibly precise to ensure everyone involved, maybe across dozens of hospitals over several years, does things exactly the same way. Consistency is paramount.
What absolutely has to be in it?
Everything, basically. The background and rationale, the specific aims and objectives, the detailed methodology, who are you recruiting, what are the exact inclusion and exclusion criteria, how will interventions be delivered, how will outcomes be measured, the sample size calculation justification, the statistical analysis plan.
Wow, okay. Detailed.
It needs to be detailed enough that, in theory, another research group could pick up your protocol and replicate your study exactly. That's the standard.
And you had a great tip. imagine writing the final paper before you start.
Yes. Try sketching out the tables and figures you expect to have in your results section. What data will go in them? Doing this often highlights gaps in your planned data collection before it's too late. You might realize, oh, hang on, to do that subgroup analysis, I actually need to collect data on smoking status or whatever it might be. It prevents mistakes down the line.
Can we look at that Sunrise trial example again? How did its protocol show this rigor?
Right. Sunrise looked at special negative pressure dressings after emergency stomach surgery, which is a high-risk group for infection. The protocol was super specific. It aimed for 840 adult patients undergoing specific types of emergency laparotomy incisions. Randomization was 1.1, computer-generated. The primary outcome was very clearly defined. Surgical site infection, SSI, within 30 days, using standard international definitions. The target was to see if the dressing could reduce SSI from an expected 25% baseline down to 15%.
So very precise targets and definitions. No ambiguity.
None. If the protocol had been vague, or if sites interpreted inclusion criteria differently, the results would have been meaningless.
How is all this data managed nowadays? It must be huge for a trial like that.
Technology helps massively. Secure online database packages like REDCap are very common now. They allow researchers at different sites to enter data electronically, often in real time.
REDCap.
Yeah, it's a widely used platform designed specifically for clinical research data capture. It has built-in checks, audit trails. It helps ensure data quality. Which links to quality assurance, you need standard operating procedures, SOPs, for data entry, regular training for staff, checks for accuracy.
And these systems help with global collaboration, too. You mentioned Sunrise involved the UK and Australia.
Exactly, because the database, REDCap in that case, and the randomization system were centralized and web-based, a researcher entering data in Sydney was using the exact same interface and following the exact same rules as someone in Liverpool. It makes managing international trials feasible in a way that just wasn't possible with paper forms.
Okay, data handled. Now, the bureaucracy, approvals, sponsorship, peer review, ethics. Sounds like a hurdle course.
It can feel like it, but it's absolutely non-negotiable. First, sponsorship. Every research study needs a formal sponsor. This is usually the lead university or hospital trust. They take ultimate legal responsibility for the study's initiation, management, and financing. They're the ones ensuring it meets all the standards.
So they're kind of the guarantor.
In a way, yes. Then peer review. Before you even think about ethics, your detailed protocol usually needs to be reviewed by independent scientific experts, often within your own institution or network. They check the scientific quality, the methodology, the feasibility. It's a crucial quality check.
Then ethics. This is the big one everyone knows about.
Yes. If your research involves NHS patients in the UK, their tissue, their data, or even their carers, you must get approval from an NHS Research Ethics Committee, or REC.
How does that work?
There's a standardized online system called IRAS, the Integrated Research Application System, where you submit your protocol, patient information sheets, consent forms, and everything else. It's complex, but centralized.
Any tips for dealing with ethics committees?
The main one is, if they invite you to attend their review meeting, absolutely go. It's much easier and quicker to clarify points and address concerns face-to-face than through weeks of back-and-forth emails.
Good tip. Beyond ethics, any other approvals?
For interventional trials, especially those testing drugs or medical devices, there's usually regulation by a national body like the MHRA, Medicines and Healthcare Products Regulatory Agency in the UK, or the FDA in the US. They look at safety and regulatory compliance.
Okay. And then there's registration. You mentioned this earlier.
Mandatory public registration. This is now a non-negotiable rule for almost all clinical trials. Before you recruit your first patient, you must register the trial's existence and key details on a public database, like ISRCTN or clinicaltrials.gov.
And journals enforce this.
Absolutely. Major surgical journals, and indeed most medical journals, will simply refuse to publish the results of a trial if it wasn't pre-registered. Full stop.
Why the strictness? What's it preventing?
It's primarily to prevent publication bias, specifically selective reporting. The worry was that researchers might run trials, get results they didn't like, maybe negative or inconclusive results, and just quietly shelve them, never publishing.
So only the good news studies get published.
Exactly. Which skews the overall evidence base, making treatments look better than they really are. Pre-registration means the trial's existence is on public record from the start. So if the results never appear, that gap is visible. It promotes transparency and accountability.
Makes sense. And all this regulation sits under a banner of research integrity.
Yes. The text mentions the Concordat to support research integrity in the UK, which outlines key commitments for researchers, institutions, and funders. It boils down to 4 core principles.
Which are?
1. Honesty in all aspects of research, including reporting results accurately. 2. Accountability, taking responsibility for the work and being transparent. 3. Fairness, treating colleagues and participants with respect, proper citation, etc. And 4. Good stewardship, using resources wisely and protecting participants.
And the absolute bottom line for anyone listening, planning a project.
The golden rule. Do not start your study, don't recruit anyone, don't collect any data until all the necessary approvals, sponsorship, ethics, regulatory registration, are fully in place and you're confident you can meet those integrity standards. You cannot get these things retrospectively. Trying to do so is unethical and potentially illegal.
Right. We've navigated the design, the protocol, the approvals. We've run the study, collected piles of high-quality data. Now, the part that often makes surgeons, maybe me included, a bit nervous, statistics.
It can seem daunting, definitely. But the single most important piece of advice, and we've touched on it, but it bears repeating, get a statistician involved early.
Not just at the end when you have the data.
Absolutely not. They need to be involved right back at the design stage. They'll help you refine the question, determine the correct sample size, and plan the appropriate statistical tests before you collect a single data point. They are preventative medicine for your research project.
Okay. Message received. Let's start with basics. Describing the data we've collected. Mean, median, mode. Refresh our memory.
Sure. The mean is just the simple average. Add everything up, divide by how many there are. The median is the middle value when you arrange all your data points in order. If you have an even number, it's the average of the 2 middle ones. The mode is simply the value that appears most frequently. And the range is the difference between the highest and lowest values.
Why do we need median and mode if we have the mean?
Because the mean can be heavily skewed or distorted by outliers' extreme values. Think about income, for example. A few billionaires pull the mean income way up. So the median income, the value for the person exactly in the middle, often gives a better picture of what a typical person earns. Same applies to lots of clinical data.
Like length of stay. Most people stay a few days, but 1 or 2 might stay for months, pulling the average up.
Exactly. In that case, the median length of stay is often more informative than the mean. Which leads us to a really critical distinction for choosing statistical tests. Is your data normal or non-normal? Parametric or non-parametric?
Okay, what defines normal distribution?
Think of the classic bell-shaped curve, symmetrical. The mean, median, and mode are all roughly in the same place at the peak of the curve. Lots of biological data, like height or blood pressure in a large population, tends to follow this normal distribution. If your data looks like this, you can use parametric statistical tests.
Parametric tests.
Like the t-tests. If you have numerical data, like blood pressure, that is normally distributed, and you want to compare the means of 2 independent groups, for example, treatment versus placebo, you use an unpaired t-test.
And if you were comparing measurements in the same group before and after an intervention?
Then you'd use a paired t-test, because the measurements are related.
But you hinted that lots of surgical data isn't normally distributed. Things like tumor size or complication counts. Why not?
Because often these things aren't symmetrical. Most patients might have 0 or 1 complication, but a few might have many that skews the data heavily to one side. Or most tumors might be small to medium, with a few very large outliers. The bell curve just doesn't fit. This is non-normal or skewed data.
So if my data is numerical but skewed, like length of stay or maybe blood loss, I can't use a t-test.
Correct. Using parametric tests on non-parametric data gives you invalid results. You must use non-parametric tests instead.
Okay, what are the non-parametric equivalents?
For comparing 2 independent groups, like the unpaired t-test, the common non-parametric version is the Mann-Whitney U-test, sometimes called Wilcoxon-Ransom. For comparing related measurements in the same group, like the paired t-test, you use the Wilcoxon signed rank test.
Mann-Whitney U and Wilcoxon signed rank.
Got it.
What about data that isn't numerical at all? Like yes, no outcomes, did the patient get an infection, yes or no, readmitted, yes or no. That's categorical data.
Right. For comparing proportions or frequencies in categorical data between groups, the standard test is the chi-squared test, or Fisher's exact test if numbers are very small.
Okay, choosing the right test is crucial. Now, interpreting the results. We always hear about p-values. P less than 0.05 means significant. What's the real story there?
The p-value is the probability of observing a difference as large as or larger than the one you found, if there was actually no real difference between the groups. This assumption of no difference is called the null hypothesis.
So p 0.05 means?
It means there's less than a 1 in 20 chance that the difference you saw was just due to random luck or coincidence, assuming the null hypothesis is true.
Yeah.
So convention says if p is less than 0.05, we reject the null hypothesis and conclude the difference is statistically significant.
But the text advises looking closely at the confidence interval, CI, as well, maybe even more so than the p-value. Why?
Because the p-value just gives you a yes-no answer on significance. The confidence interval gives you much more information. It provides a range of plausible values for the true difference in the wider population.
How does that help?
Let's say your trial finds a new drug, reduces blood pressure by an average of 5 mmHg compared to placebo, and p is 0.03, so significant. That sounds good. But what if the 95% confidence interval for that difference runs from, say, 0.5 mmHg to 9.5 mmHg? That tells you the true average reduction in the population could plausibly be as small as 0.5 mmHg, clinically tiny, or as large as 9.5 mmHg, maybe quite useful. It gives you the precision or uncertainty around your estimate.
And what if the confidence interval includes 0?
Ah, that's the key point.
Yeah.
If the 95 per CCI for the difference between two groups includes the value 0, it means that no difference is a plausible value for the true effect. In that case, even if the average difference looks promising, the result is usually considered not statistically significant at the 0.05 level.
So the CI gives you both the size of the effect and the statistical significance in one go.
Essentially, yes. It's a much more informative measure than the p-value alone. You see the potential range of the true effect.
There's a warning here, too, though, about looking at too many things. P-hacking.
Yes, be very wary of studies that test dozens and dozens of secondary outcomes or subgroups. If you run enough statistical tests purely by chance, some will come up with p 0.05. It's called the multiple comparisons problem.
So you might find a significant result just by luck.
Exactly. Good studies pre-specify their primary outcome and limit the number of secondary analyses. And a good rule of thumb is, if you need complex statistical gymnastics to demonstrate a difference that isn't readily apparent clinically, then the clinical significance of that difference is probably quite low, even if the p-value squeaks under 0.05.
Okay, we've designed it, run it, analyzed it carefully. We have results. The final hurdle, getting it out there, dissemination.
Right.
And the message seems to be, publish it, even if it's not groundbreaking.
Absolutely. The text is very clear. No project is worthless, even if the results are negative or inconclusive. There's a strong ethical imperative to publish.
Why is reporting negative results so important?
Because failure to do so leads to publication bias. If only studies with positive, exciting results get published, the overall literature becomes systematically biased.
Yeah.
It creates an overly optimistic picture of how well treatments work. We need the full picture, warts and all, to make properly informed decisions. So yes, publish those non-dramatic findings too.
Okay, assuming you're going to publish, choosing where, how do you decide?
You need to match the study to the audience and the journal scope. A major multi-center RCT that could change practice globally. Aim for a top-tier international journal. A smaller local audit or a very niche case series. Perhaps a regional surgical meeting presentation or a more specialist journal is appropriate. Be realistic.
And when you've chosen your journal, the submission process. Any top tips?
The single most important piece of advice sounds boring, but it's crucial. Read and follow the journal's instructions for authors meticulously. Word counts, reference style, figure formats, abstract structure, get it wrong, and the editorial assistants might reject it before it even gets to a reviewer. Don't fall at that first hurdle.
We're also seeing more open access journals now.
Yes, the model is shifting. Traditionally, journals made money from subscriptions. In the open access model, the author, or their institution funder, pays a fee upon acceptance, and the published article is then freely available for anyone to read online immediately. It changes the economics, but increases visibility.
Regardless of the journal type, the structure of the paper itself seems pretty fixed. The IMRAD format?
IMRAD is inviolable. It stands for Introduction, Methods, Results, and Discussion. It's the universal standard for scientific papers because it provides a logical, predictable structure for readers.
Let's quickly break down what each section should achieve. Introduction.
Keep it short and sharp. Briefly state the clinical problem or context. Summarize what's already known and where the gap in knowledge is. Then clearly state the specific aims or objectives of your study. What question are you answering?
Methods. Sounds like it needs detail.
mapping detail here. This is the how we did it section. Describe the study design, the setting, the participant, exclusion criteria, the interventions, how outcomes were measured, the sample size calculation, and the statistical methods used. Identify potential biases and how you tried to minimize them. If you used a new technique, describe it fully. If it's standard, just reference it. Enough detail for replication.
Results. Focus on visuals.
Yes. Use tables and figures effectively to present the main findings clearly and concisely. The text shouldn't just repeat everything that's in the tables. It should highlight the key results and draw the reader's attention to the most important data points. Be objective. Just state what you found.
Okay, and finally, the discussion. This sounds like the hardest part to write well.
It often is. This is where you interpret your findings. What do they mean? How do they compare to what other studies have found? If they're different, why might that be? Discuss the strengths and, importantly, the limitations of your study.
And suggest next steps.
Yes, suggest implications for clinical practice, cautiously, and what future research might be needed. But two big don'ts. One, do not just repeat the introduction or summarize the results again. Assume the reader has read those sections. Two, avoid what the source calls flights of fantasy.
Meaning?
Don't over-speculate wildly about the potential future impact or applications far beyond what your data actually supports. Stick to interpreting the evidence you have generated. Grounded interpretation, not wild imagination. Editors hate unwarranted hype.
Okay, so that's how we write it. How do we, as readers, evaluate the papers that land on our desks or in our inboxes?
Use the tools. There are established critical appraisal checklists designed specifically for this. For RCTs, the main one is the CONSORT statement and checklist. For systematic reviews and meta-analyses, it's PRISMA.
What do these checklists do?
They list all the essential items that should be reported in a high-quality study of that type. Things about randomization methods, blinding, follow-up rates, statistical analysis details. If a paper doesn't report these key methodological points clearly, you should be skeptical about its quality. There are even scoring systems, like the JDAD score for RCTs, that try to quantify trial quality based on reporting of randomization and blinding.
So this whole rigorous process from question to publication and appraisal feeds into the bigger picture of evidence-based surgery.
Yeah, exactly. It reflects a fundamental shift, moving away from surgery being just an art based purely on individual experience or opinion. This is how I've always done it. Towards clinical practice that is explicitly based on the best available scientific evidence generated and evaluated using these rigorous methods we've discussed.
And organizations like Cochrane are key here.
The Cochrane collaboration is hugely important. They produce systematic reviews, exhaustive searches, and appraisals of existing trials on specific topics to provide the most reliable summaries of evidence. Their library is a vital resource.
And meta-analysis, that sounds complex.
It's a specific statistical technique used within some systematic reviews. It mathematically combines the numerical results from multiple individual studies, if they're similar enough, to produce a single overall estimate of the treatment effect. This pooled result is often much more precise and powerful than any single study alone. It's about synthesizing all the relevant evidence. #tag outro.
Right. So we've really covered the waterfront today. From that crucial first step, is it audit or research, using the HRA tool, through the formal 7-step audit cycle for quality improvement. The rise and strategic importance of collaborative research, like the WMRC model. The absolute necessity of a rock-solid protocol, getting the sample size right.
Yeah, avoiding those type I and 2 errors.
Choosing the correct statistical tests, understanding p-values versus confidence intervals. And finally, the ethical imperative to publish, using that standard IMRAD format. It really is the full playbook.
It is. You should now have that framework to approach any project or critically read any paper with a lot more clarity and confidence.
And if we zoom out, thinking about the big picture, you mentioned how things like collaboratives and mandatory trial registration are changing the field.
I think they really are.
Yeah.
This move towards more collaborative, transparent, globally registered research, it's starting to smooth out those old variations in practice that were based purely on anecdote or local tradition. Well, when large, well-conducted studies provide clear evidence, it becomes harder for individual clinicians or units to justify doing things completely differently without good reason. The evidence points towards the best way of managing patients becoming clearer, less subjective. It drives standardization towards best practice.
Which brings us to our final provocative thought for you, the listener, to mull over.
Building on that idea of standardization, think about your own area of surgical practice. What specific aspect, maybe it's how certain drains are used post-operatively, or a particular technique for wound closure, or maybe a specific post-op pain relief regimen. What area do you think is still largely treated as an art, based more on individual surgeon preference or historical habit than on solid comparative evidence?
Yeah, where's that lingering subjectivity?
Exactly. Which of those art forms do you think most urgently needs the structure and objectivity of something like a multi-center snapshot audit, or even a full RCT, to really settle the debate and establish a clear, evidence-based consensus?
Something to ponder on your next ward round, perhaps.
Definitely.
Yeah.
Think about it, analyze your own environment, and keep digging into the sources. Thanks for joining us for this deep dive.
We'll see you next time on the